everyone, my name is Dr. Monica, working as Associate Professor in NIET Pharmacy Institute, Greater Noida, which has been affiliated by Dr. APJ Abdul Kalam Technical University, and I am going to take the subject, that is Pharmaceutical Microbiology, having code BP 303T. Now, what topic I am going to cover in this lecture, that is study of different types of microscopes. Now, microscopes like phase contrast microscopes, dark field microscopes, bright field microscopes, we have even electron microscopes today. In the recent world, we can easily go for SAM and TAM, that is scanning electron microscopy, and that is transmission electron microscopy. So, but in this lecture I will mainly focus on the instruments which are mainly used within the labs for the research purpose, microscopes which are being used like bright field, dark field and phase contrast, which are easily available in the pharmaceutical labs. So, how mainly what is their construction part, what is their principal part, how these instruments actually work. So, mainly we will deal with the microscopy today in this lecture. That contents which I am going to cover in this lecture are the light field or optical microscopy. This is being further divided. Light microscopes we can further divide into various parts like bright field microscopes, like dark field microscopes and phase contrast microscopes. So, these three microscopes comes under optical microscopes or light microscopes, but we just have these only microscopes available with us. We are having even the that is advanced version of the microscopes too, like if we go for microscopy, microscopy actually deals with the study of microscopes, actually deals with the study of various microscopes available to us as what is the needs of studying this microscopy, as we can't see microbes without the microscopes. If we don't have any microscopes, we won't have generated any of the microscopes, we won't be able to even see the microorganisms, we won't be able to differentiate between bacteria, fungi or viruses. For that we actually need microscopes, and that is why this topic has been put under this course of that is pharmaceutical microbiology. We need to study few of the microscopes. Now, what are those microscopes actually are? We can categorize our microscopes into optical microscopes or electron microscopes. Optical means they require the source that is light. Light is always the source, that is why they are also known as light microscopes, and under light microscopes have further been categorized into three categories. One is bright field microscopes, second one is dark field microscopes, and third one are phase contrast microscope. This we will study in this lecture, and rest of the part, that is electron microscopes containing SAM, TAM, I will try to cover in my upcoming lecture, that is what actually electron microscopes and what are the advanced versions of electron microscopes like scanning electron microscopes and transmission electron microscopes. In this lecture I will just only deal with the light microscopes. These light microscopes are also available in the normal labs. We can easily see the normal microscopes which are generally available in the 12th class or 11th class, you must have seen. Even you must have seen in some of the pharmaceutical labs also, that normal microscope we actually name it as bright field microscopes, and the advanced version of bright field microscopes are dark field microscopes and phase contrast microscopes. Now, what actually the principle of bright field microscopes and how it works, what are their construction parts, what is their working, what is their procedure, how we can analyze our sample using the microscopes, and what advances have been made within this dark field and phase contrast to overcome the limitations of bright field, that we will deal in this lecture. So, first of all, bright and dark field microscopy. Now, what we actually mean by bright field microscopy? Bright field microscopy is the conventional technique. Conventional, we are using this technique from even the 1950s. If we see, even in 18s also we were using this simple microscopes which in which we just place a slide and after adding the specimen on the slide, we observe under the microscope, and we can easily see the structure of plant cells, we can easily see the structure of animal cells, we can go for the study of various bacteria, various that is viruses, various fungi. So mainly these are suitable for observing the natural colored. We can't see with the help of simple conventional bright microscope the transparent images. If we want to if we go for the simple bright field microscopes, we can only observe the colored images. This is the main limitation of bright field microscope, that we won't be able to see the transparent images until unless we have not stained it, we have not added any of the staining reagent within those samples, the cells which are transparent in nature. If we have transparent cells, we won't be able to observe because light directly passes from the slide, and if we have transparent cells or specimens we want to see that are transparent in nature, that will be not be seen within the eyepiece. We won't be able to observe those images if they are transparent in nature. For that we need staining reagents, we need to add some of the dyes, some of the reagents, so that they can color the cells, or colored cells we can easily observe with the help of bright field microscopes. So bright field microscopy is conventional technique. It is suitable for observing the natural colors of the specimen, colored specimens we can see, but if they are transparent, we need to if we want to observe those cells which are transparent, we have to stain those samples, that samples must be stained. The microscopic field is brightly lighted and the specimen appears dark. What will happen? And bright field microscope, if we what we need to do, first of all, when we want to observe anything or any of the cells or any of the species or any of the cultures under the microscopes, first we need to make a slides we have to make. In slides what we need to do, we have to add those samples which we want to observe, and after adding those samples we cover it with the help of cover slips, and after that we will place those samples on the if you see this microscope, we always place our samples on this stage. This what will happen, these are the various parts of a simple microscope. If we have seen in the lab, this is a simple microscope, it has a light source. Bright field normal microscope will not contain any light source. You have to go, you have to see under sun rays, or you have to find out a space where you can keep your microscope, and sun rays pass through the specimen. But the advanced version was bright field, in which there was a light source. Light source was attached in a bright source or bright field microscopy. This is a light source. This light source will pass through the condenser, and from condenser or diaphragm it reaches the specimen which has been placed here, and after reaching it the rays will move towards the objective, and from objective we can see our image within the eyepiece. Eyepiece will gives us the image of that particular species. And even we have objective lenses which are of various that is magnification like 40x, 10x, 100x, depending upon the lens we are going to see or image we are going to see, we can even change the objective magnification, that is 10x, 100x or 40x. So this will gives, what will happen, light source, light will move towards the condenser, and from condenser it will condenses to the glass slide, and from glass side we can easily observe this light will pass through the objective lens and it will reach the eyepiece, and we will be able to see we will be able to observe the image. But if the image is transparent, the whole light will pass through the glass slide, and image is also similar to that of a glass slide, that is also transparent in nature. We won't be able to see or differentiate that image with the help of eyepiece. So that's why bright field microscope will not work for the images or for the species which are that is transparent and which are not colored. So that's why we move forward towards the dark field, because we need to add the staining reagents within the slide, and that may make the dissolving power to reduce. We won't be able to resolve the organelles within the cells. Cells also contain some of the organelles like mitochondria, that may contain nucleus, that may contain cytoplasm, they contain plasma membrane, we won't be able to resolve those organelles within the cells. We can see the structure, we can see the that is image of that particular cells, but resolving power reduces because of adding some of the staining reagent. And even the bright field also have itself also have some less resolution power, it does not have much more resolution, it can only gives us the structure of the cells, it will let us know the shape of the cells, but bright field microscopy will won't work if we want to resolve the organelles within the cells, if we want to see the organelles within the cells. It will gives us just the shape of the cells, but then also we need to add some of the staining reagent if they are transparent in nature. So for bright field, that means, as you can see in this image also, that this is a bright field image in which the structure, or if structure is colored, then we can see the structure. If this structure is also colorless, then image will not be created within the slide, and we won't be able to differentiate the cells and the glass slide. So transparent images can't be seen through this bright field microscopy, as well as we can't be able to differentiate this. It has less resolving power. That is the basic limitation of bright field microscopy, that it has that is resolving power is very less of bright field microscopy. That is why the scientists, the researchers move towards, they try to work on it, that how we can see the organelles within the sample, how we can differentiate, that is we can see the transparent cells. To observe the transparent cells, if we add staining reagent, they may damage the cell, they may that is stain the organelles, they deteriorate the image actually when we add some of the staining reagent within the cells. We won't be able to see the live cells as it is they are within the nature. So if we want to see natural or transparent cells, so then we have to move towards the dark field. Scientists develop a new version of bright field, that is known as your dark field. Now what dark field will do? Dark field is a special type of instrument in which they have add on one addition to the microscope. That addition was the opaque disc was added to the, how opaque disc will work. Dark field microscopy, the microscopic field is dark. What will happen, as you can see even in this image, what will happen, your whole slide, glass slide will be dark, the background will be dark, but the cells will remain as it is. You will be able to see even the transparent cells, transparent specimens, which will appear brighter with the light, but the whole that is slide will become, background will become dark. So in bright field we were having background that is transparent, the background was that is colorless, whereas in case of dark field, what will happen, your field background will become dark. As you can see, the whole background is dark, but the image will be brighter image you will get. In bright field we were getting brighter image, we were getting brighter slide, the whole light was passing through the slide and we were not able to see the transparent cells. But in this case, in dark field, what will happen, your field becomes dark. As you can see even in this image, that field becomes dark and cells you can easily see in the natural form, whether they are transparent, whether they are having colorless organelles are there within the resolving power of your sample will increase using the dark field microscope. So this is accomplished using a specially sized disc, specially sized opaque disc was attached to the instrument. Now where that disc or where that opaque disc or patch was attached to block the light, so that light will not be able to pass through the whole slide, light will only pass through the image, light will not pass through the slide, but light will only pass through the image, so that your image looks brighter but the whole background looks darker. Light will be focused, light will be condensed in such a form that light will just reaches the specimen. It will not pass through the glass slide, your whole background will be dark, the light will just pass through the image and your image will look brighter in nature. So that is why a patch, a stopping block was attached in the that is path of light. So, how you can differentiate? You can easily see this slide. You can see that is, this one is bright field, this is the image of a bright field, this one is the image or principal part of a, that is dark field, and even the phase contrast microscopy. So, these dark field microscope and phase contrast microscopes are the higher versions of that is light microscopy. Higher versions, how that is they will increase the, we will be able to see the live cells, we do not need to add any of the staining reagents within the sample. These are the advanced version, they have been designed in such a way that we will be able to see the transparent objects, transparent images, live cells on the slide, but on a dark background, the whole background will become dark and you will be just be able to see that particular species. Now, how that is going to happen? One is, as you can see in this, one opaque disc, an extra addition was added, that is before the condenser, below the condenser and above the light source. As you can see, there was a light source, there was condenser lens, there was specimen. Here we have played the specimen, and from the specimen when light passes through the objective lens, it reaches the eyepiece, and from eyepiece we observe the samples. But in this case, what will happen? We have blocked the light using a opaque disc. One opaque disc is there, dark disc having dark that is the opaque disc that will block the light. Dark disc will be there that will block the source of light, and when light will pass, it will condenses out in such a way that will only reaches the specimen. It will not pass through the whole slide, it will just pass through the, that is specimen. It will not cover up the glass slide, it will not pass through the glass slide, it will be condenses in such a way that it will just pass through the specimen, and you will be able to see the specimen as within the dark background, but the background becomes dark, the background will become dark and you will be able to see the that is image. So, this is what we can see. This one was the dark field microscope in which one extra disc was attached, that disc was known as opaque disc, and one more higher version of that is dark field is your phase contrast microscopy, in which further 2 additions were done. In this, only one addition was done, that is one opaque disc was attached, that is before the condenser lens and above the light source. But what happened in phase contrast microscopy? They attach 2 extra annular disc. One is annular diaphragm was added, and second one is phase contrast or phase contrast diffraction phase plate was added. 2 extra additions was done in case of phase contrast microscopy, so that the resolving power of the bright field increases. From bright field, scientists move towards the dark field, and from dark field they move towards the phase contrast microscopy. So, just to enhance their resolving powers, to enhance their that is structural image, we must be able to see the organelles too. So, that is why they move from bright field to dark field, and from dark field to that is phase contrast microscopy. In dark field, only one extra addition was done, whereas in phase contrast there was 2 extra addition was done. One is that is annular diaphragm that was placed in between the light source and the condenser, and the diffraction plate which was added in addition to above the objective lens and before the ocular lens. So, now phase contrast microscopy. Now, what is what actually done in case of phase contrast? Mainly to enhance the, that is what we call as resolving power, and to enhance so that we can easily see the transparent images. So, scientists move towards prefer to use the phase contrast microscopy, in which 2 further additions were done. In phase contrast microscopy, based on even the refractive index changes, we can differentiate between the organelles. We will be able to differentiate between the structure of cells or cell membrane, plasma membrane. Further, we can easily observe the organelles within the cell structure, that is mitochondria, cytoplasm, nucleus will be easily visible based on their changes in the refractive index. Phase contrast microscopes are the microscopes which can even note down or which can even resolve the that is organelles too. which are spaced very, there is very little space between those organelles. That organelles will be visible using the phase contrast microscopy. Resolving power will increase due to the addition of phase contrast, that is diaphragm or phase contrast annular plate which was added. Though those two addition just made the phase contrast microscopy, even we were able to see the live cell, we were able to see the, that is resolve the organelles using the phase contrast microscopy. Now who is the scientist who developed that phase contrast microscopy in 1934 was a Dutch physicist, that his name was Frits Zernike. Now what he did, phase contrast microscopy actually is very extremely valuable to study the biologicals or to study the cell structures, to study the organelles within the structure, but main that how, on what principle it works, it also creates a dark background, but your light will just pass through the, that is specimens, your specimens will be bright, whereas the whole background will be dark, as was seen in case of dark field. But as extra annular, that is refractive, based on refractive index, phase contrast plate was also added, so that was able to recognize the differences between the refractive index, diffraction pattern, constructive patterns were be able to be noted down using that annular phase contrast plate which was added before the ocular lens but above the objective lens. So two additions were done in phase contrast microscopy to observe the unstained cells and to observe or differentiate or to increase the resolving power of the image. So this special optical system makes it possible to distinguish even the unstained cells, and which also have on base, mainly based on their refractive index changes. If there is any change within the refractive index, if there is any change in the diffraction pattern, constructive patterns, that microscope will be able to analyze, that microscope will be able to differentiate. Now what are the main parts which were attached along with the construction part of the normal microscopes, conventional microscope? It was also having, that is condenser lens, it was having that is specimen plate, stage was there, it was having, that is objective lenses, it was having ocular lenses, it was having eyepiece, but along with that it was having two additional that is construction materials or construction pieces, that were, first one is annular diaphragm was attached, as you can see in this image, that is this one, as you can see there will be light source, but one extra addition was this, that is annular diaphragm which was attached before the condenser but after the light source. This annular diaphragm was added. Here we will have to place the specimen, here is the objective lens, and but extra attachment is here, that is phase plate. These two additional that is plates were added within the normal microscope. One is annular diaphragm and second one is phase plate was attached, that is before the eyepiece but after the objective lens. In between objective lens and eyepiece, this extra phase contrast plate was attached, which was able to differentiate or which was able to analyze the changes in refractive index in the diffraction patterns. So the why that annular diaphragm was attached, first it is situated below the condenser, it is made up of circular disc having circular annular grooves. Always annular grooves are there within the, that is annular diaphragm, that grooves will allow the pass of light, that light will pass, but that light will only be passed through the grooves. The whole disc will be dark, the whole annular diaphragm will be dark, but having but it will have some of the grooves through which light can pass, and that light will just condenses with the help of condenser to the specimen. That will not pass through the whole slide, it will just pass through the specimen, and so that your specimen will be that is the specimen must be brighter and the whole background must be dark. After that, the phase plate. The phase plate, it is either negative phase plate or it may be positive phase plate. It is thin, it is having a thin or thick areas even in the place they have some of the thick areas, some of the thin areas to recognize the differences in refractive index. So, plate was designed in such a way that you must be able to differentiate between the that is refractive indexes. So, that phase plate is transparent disc. With the help of annular diaphragm and the phase plate, the phase contrast is obtained within the microscope. With the help of phase contrast plate, the contrast will generate and the refractive changes in refractive index will be able to be noted down, and you will be able to differentiate, that is based on the refractive index, that is various cell components, that is organelles within the cells you will be able to see. So, partially what is the main procedure, how the light pass? Partially coherent illumination produced by the tungsten halogen lamp. The halogen lamp which is placed here, the light will pass, that light will move from this annular diagram. This is a groove from which, this is a dark place, this one is also dark, but this is what is known as your groove. From with this groove, light will pass on, and light will pass through the grooves within the plate. Those light will be condenses, and those light will after condensing, this will pass through the specimen, and from specimen they reaches the objective, and from objective here we have plate which is from somewhere there it will be thick, from somewhere it will be thin, depending upon, so that it may that is it will collect all the light which is coming through the objective lens. Here it will be focused, and that based on their change in refractive index, it will be able to resolve even the organelles within the samples. So, as you can see even in this, that is slide, you can easily see, you can observe the differences, how the image will look within the bright field. This is a bright field in which the transparent images are there. You won't be able to differentiate those transparent images. This is dark field. As the background is dark, the cells are transparent in nature. So, easily you can, light is only passing through the specimen, you can easily observe the specimen on the dark background will be observed. In phase contrast microscopy, there is a phase plate. So, refractive indexes will be able to change in refractive index will be able to, you will be able to analyze. So, even you can see the organelles within the cell, nucleus, you can easily observe the nucleus within the cell, you can easily observe the cytoplasm within the cells, you can easily observe the differentiate between the cell membrane or plasma membrane within the structure using the phase contrast microscopy. And even advanced versions, higher advanced versions are also there, like fluorescence. This colored images you can observe, that is what is the color of a nucleus. As you can see in this, here it is a nucleus, here it is a cell, flagellas are there. So, you can easily observe, differentiate between organelles, you can see the color difference, even in the difference in the color. So, these are the application, these bright field and dark field are used for that is to view blood cells, for identification of bacteria. They are used to differentiate between various types of algae, identify hairlines. They are used to check diamond and other precious stones. These are used to differentiate between the mounted cells and tissues. We can see the live bacteria. We can even determine the motility of bacteria within the culture. We can easily analyze the motility in the culture, motility of bacteria within the culture. We can use this phase contrast microscope or light microscope, advanced version of bright field microscope for the high contrast images. We can easily see the transparent images, we can see the living cells, we can observe the microorganisms, but in their natural form, not by staining them, or in the natural form we can easily observe even the transparent microorganisms, transparent cells. We can have a thin tissue slices can be observed, we can have lithographic patterns, we can observe the subcellular organelles including even nucleus we can see using that is phase contrast microscopy. And this phase contrast microscopy can further be used for various biological research purpose. For numerous biological research it is allowing us to do various researches, because we are able we can even see the organelles within the cells. This is highly efficient tool or efficient instrument by which we can do the further research, biological research too. And these are the references which you can follow, and like Prescott has given industrial microbiology. You can go for fungi, viruses, bacteria. You can follow these references which have even AKtU site. You can see that various references have been given. So, you can follow these books for for studying. Even Pelzar is the best one. You can go for Pelzar if you want to study the microscopic techniques. So, thank you so much. Thanks a lot.